nucleic acids are the building blocks of life. Wait, but isn't DNA the building blocks of life? Yes, both are correct. DNA is a type of nucleic acid, and in this video we are going to have a quick look at another type, RNA. We have both DNA and RNA in our bodies. We need them both. DNA is the blueprint. It contains all of the instructions for the cell to grow, function, and replicate. The RNA carries out these instructions. It copies and transfers the genetic code from the DNA to ensure the relevant proteins are made. So just think of it as DNA makes RNA makes proteins. So let's look at how they differ. Whereas DNA stands for deoxyribonucleic acid, RNA stands for ribonucleic acid. Whilst DNA is double-stranded, RNA is single-stranded. Like DNA, RNA is made up of a long chain of nucleotides. Each nucleotide consists of a ribose sugar, phosphate group, and nucleotide base. RNA has a sugar called ribose, whereas DNA has a sugar called deoxyribose. Look here, RNA has a base, uracil, or U, whereas DNA has the base thymine, or T. So in RNA, C and G still pair, but now A and U pair. We need to know about RNA because we will see it when learning about protein synthesis. We will see these two special types of RNA, messenger RNA, which is known as mRNA, and transfer RNA, which is known as tRNA. We will see the mRNA being synthesized inside the nucleus, copied from the DNA code. The tRNA is found in the cytoplasm. For many years, we just thought RNA was a DNA photocopier, as mRNA, the protein builder, as tRNA, and found in ribosomes as rRNA. However, RNA can also act as enzymes to speed up chemical reactions, and in many viruses, they have RNA instead of DNA. The RNA carries the genetic codes in those viruses. So there we have RNA. From this video, you should now know about the existence of RNA. It is a single-stranded nucleic acid that has a base U instead of a base T.
Okay, so the topic of this short video today is going to be the three different types of RNA: mRNA, tRNA, rRNA. So let's go ahead and get started. Okay, so let's talk about ribonucleic acid. When you look at the picture, RNA on the right, you can see it's only a single strand of nucleotides. But the picture of DNA on the left, DNA is a double helix because it's made from two strands of nucleotides. Well, RNA is still a nucleic acid, so it's still made from building blocks called nucleotides. And nucleotides have three parts, one being a phosphate, a second being a sugar molecule. Now, the picture that just showed up, this is the sugar called ribose. This is the sugar of RNA. On the left, this is the sugar called deoxyribose. This is the sugar of DNA. They're virtually identical. Highlighted in red is the difference. The sugar on the left, deoxyribose, it's been deoxied. It's missing an oxygen atom. Other than that, they're very similar to one another. And then the third part of a nucleotide is the nitrogen base. In RNA, the nitrogen base might be A for adenine, it might be G for guanine, it might be C for cytosine, and it might be U for uracil. These are the four nucleotide nitrogen bases of RNA. And ultimately, we're going to learn about the three kinds of RNA. We're going to learn about mRNA, and then we'll learn about tRNA, and then we'll learn about rRNA. So let's talk about mRNA first. Okay, so the M of mRNA stands for messenger RNA. And you're going to see it's going to start in the nucleus of a cell, but it will end up at a ribosome. Well, here's a cell right here. And let's talk about eukaryotic cells. First, eukaryotic cells have a nucleus, and in that nucleus is the DNA of the cell. Now, you might know that DNA is the instructions or the blueprint to build all the proteins that we need. But the actual maker of the protein is a ribosome. A ribosome is the actual builder of the protein, and that's out in the cytoplasm. So what does mRNA do? It makes a copy of the DNA code. It makes a copy of the DNA sequence. It makes a copy of the DNA instructions. And messenger RNA will deliver the instructions from the nucleus to the ribosome. And once the ribosome has the instructions, what the ribosome will do is will build a protein. And it does that by gathering up a bunch of amino acids and linking them in a long chain. which eventually is a protein. And this is a process called translation. That's what a ribosome will eventually do, is gather the amino acids and link them into a protein. Okay, and now for transfer RNA. The T stands for transfer RNA. And the location is going to be in the cytoplasm. And what happens is, we said a moment ago the ribosome is going to gather amino acids to make a protein. But how? How does the ribosome actually obtain the amino acids? Well, transfer RNA will pick them up and carry the amino acids over to the ribosome. So when a ribosome is making a protein, it needs the help of transfer RNA. Transfer RNA is actually picking up the amino acids and carrying them and delivering them to the ribosome. And one by one, the amino acids are picked up by transfer RNA, delivered to the ribosome. And so you see what's happening now is the ribosomes are linking the amino acids together one by one in order to make a protein. This is, again, the process called translation. That's the general job of transfer RNA. And then the final kind of RNA is R, or ribosomal, rRNA. And rRNA is the main component of a ribosome. A ribosome is a cell part that makes proteins, but ribosomes are made from something. And rRNA will bond with some special proteins in the cell to form a ribosome. So a ribosome is actually made from twisted together rRNA and proteins. And then a ribosome, when it's constructed, it will help to build a protein through the process called translation.
Genetic information flows from DNA to RNA to protein synthesis is through a process called the central dogma. The first process is the transcription process. It is a process that copies the DNA into mRNA. This process occurs in nucleus because we can find DNA in the nucleus. Inside the nucleus are free-floating molecules that the RNA polymerase will use to mix copy. RNA polymerase is enzymes that binds into promoter sequence, and it's going to make a copy of DNA using RNA molecule in a 5 to 3' direction. You can see here that the sequence of nucleotides in the DNA is used as a template to determine the sequence of the bases in the mRNA molecule. And there is no thymine in the RNA. Instead, it is replaced with uracil. Whenever this RNA polymerase reaches into terminator sequence, it is going to unbind to the DNA, allowing the mRNA to live in the nucleus. This messenger RNA, a copy of DNA, will find the ribosomes that will be used in translation process.
In biology, transcription is the process of copying DNA into RNA by an enzyme called RNA polymerase. Generally, RNA polymerase only wants to transcribe regions of DNA called genes, as these provide the correct instructions for making useful RNA. However, the human genome is 3.2 billion base pairs long, and only a small percentage of it contains genes. So how does RNA polymerase know where to transcribe? It makes use of what are called promoters. Promoters are DNA sequences at the beginning of genes that provide a landing pad for RNA polymerase and a bunch of other proteins that altogether work to initiate the transcription process. In other words, a promoter both marks the start of the gene and helps bring it in for activation. Although promoters for different genes aren't exactly the same, they do share some common features. For instance, a good fraction of promoters contain what is called the Tata box, a sequence characterized by alternating adenines and thymines. This region of the promoter is recognized by a protein that plays a role in stabilizing RNA polymerase and encouraging its activity. In addition to the promoter, some genes make use of enhancer sequences. These are short sequences that recruit so-called activator proteins that help RNA polymerase work much more efficiently. Fascinatingly, enhancers are often located thousands of base pairs away, and so the DNA must loop around to allow for enhancers to get close to the promoter. Overall, by using enhancers and promoters as genetic signals, the transcription machinery within any given cell is able to appropriately express its genes.
RNA editing is a molecular process through which some cells can make discrete changes to specific nucleotide sequence within an RNA molecule after it has been generated by RNA polymerase. What it means is when a cell makes an RNA molecule, its sequence can be changed after it is synthesized to change the final sequence of the protein. RNA editing may include the insertion, deletion, and base substitution of nucleotides within the RNA molecule. Here, you need to understand what is insertion, deletion, and base substitution of nucleotides. Insertion means when there is an addition of a nucleotide in the sequence. Deletion obviously means when there is a deletion of a nucleotide in the sequence. Base substitution means when there is an exchange of one nucleotide with another. RNA editing has been observed in tRNA, rRNA, mRNA, or miRNA molecules of eukaryotes, as well as in viruses, archaea, and prokaryotes. RNA editing occurs in cell nucleus and cytosol, as well as within mitochondria and plastids. Alright, let's discuss the process of RNA editing. Editing by insertion or deletion are also known as pan editing. RNA editing through the addition and deletion of uracil has been found in kinetoplasts. A kinetoplast is a network of circular DNA molecules present in a large mitochondrion. Because this may involve a large fraction of the sites in a gene, it is sometimes referred as pan editing. Pan editing starts with the base pairing of the unedited primary transcript with a guide RNA or gRNA, which contains complementary sequences to the region around the insertion or deletion points. After that, the newly formed double-stranded region is then enveloped by a editosome, a large protein complex that catalyzes the editing. The editosome opens the transcript at the first mismatched nucleotide and start inserting uridines. The inserted uridines will base pair with the guide RNA, and insertion will continue as long as A or G is present in the guide RNA and will stop when a C or U is encountered. The inserted nucleotide sequence causes a translational frame shift, and because of the translational frame shift that we already discussed, the mechanism of editosome involves an endonucleolytic cut at the mismatched point between the guide RNA and the unedited transcript. The next step is catalyzed by one of the enzymes in the complex, a terminal uridine transferase, which adds uridines from UTP at the 3' end of the mRNA molecule. Ends are held in place by other proteins in the complex. Another enzyme, a uridine-specific exoribonuclease, removes the unpaired uridines. After editing has made mRNA complementary to guide RNA, and RNA ligase rejoins the end of the edited mRNA molecule. Another mechanism of RNA editing is editing by deamination. The editing involves cytidine deaminase that deaminates a cytidine base into uridine base. Also, there is another type of deamination where adenosine is deaminated into inosine. Alright, let's discuss the functions of RNA editing. RNA editing can alter the sequence, it can alter the future path of the RNA molecule, it may cause a viral genome mutation, and it may alter secondary structure of RNA molecule, causing a different protein binding site.
So the next one is the ribosome. So first, let's define what is a ribosome. So it is an intercellular structure made of both RNA and protein, and it is the site of protein synthesis in the cell. So um, the ribosome reads the messenger RNA or mRNA sequence and translates that genetic code into a specified string of amino acids. Then it will then grow into long chains that fold to form proteins.
Now, the question is, what is the role of ribosomes in protein synthesis? So, as what I've said earlier, the ribosome reads the messenger RNA or mRNA sequence and translates that genetic code into a specified string of amino acids, which it will grow into a long chains that fold to form proteins. So, a ribosome is the cellular machinery responsible for making proteins. So, there are many ribosomes in each cell, each made up of two subunits. So, getting two subunits lock around the messenger RNA or the mRNA, and it will then travel along the length of the messenger RNA molecule, which it reads each three-letter codon. So, the ribosome serves as a docking station for the transfer RNA or the tRNA na nagmatch sa sequence of bases of the messenger RNA. So, each three-letter codon sa messenger RNA pairs with the matching anticodon on a specific transfer RNA or tRNA, and that specific RNA allows for addition of a specific amino acid on the end of the growing protein chain. So, ang ribosome dayon will break up after the completion of the protein.
Welcome to Moo Moo Math and Science. In this video, I'd like to talk about the structure and function of the ribosome. When you look at the pictures of this plant and animal cell, you can see tiny dots on the ER and in the cytoplasm. These dots are the ribosomes. But why are they important to the cell? Ribosomes are small protein factories found in cells. Ribosomes are found in plant, animal, and bacteria cells. The average eukaryotic cell has approximately 10 million ribosomes. If you take a look at this picture of a ribosome, you will notice a large subunit and a small subunit, which both contain protein and RNA. In plant and animal cells, ribosomes are found floating in the cytoplasm or bound to the rough ER. Free-floating ribosomes create proteins to be used within the cell, while ribosomes attached to the ER create proteins that are either bound to the cell membrane or are used outside of the cell. Ribosomes use a process called translation to create proteins. In basic terms, here's what happens. mRNA or messenger RNA that has been created at the nucleus leaves the nucleus and travels to the ribosome. Next, tRNA then travels to the ribosome and reads the mRNA in triplets called codons. The tRNA has an amino acid attached to one end. As the tRNA reads the mRNA, the strings of the amino acids are put together in the correct order and creates a protein.
Genetic code refers to the instruction contained in a gene that tell a cell how to make a specific protein. Each gene's code uses the 4 nucleotide bases on DNA: adenine, cytosine, guanine, and thymine, in various ways to spell out 3-letter codons that specify which amino acid is needed at each position within a protein. Genetic code is the story of biology and genetics in the 19th, 20th, and 21st centuries, as well as its premises and its perils. Oswald Avery in 1944 proved that the genetic codes that DNA was indeed the carrier of heredity information, ending more than 80 years of productive speculation. But as important as DNA was to the so-called heroic era of molecular biology, spanning the generation of scientific discovery after the Second World War, and as important as DNA in the revolutionary sciences of genetics and genomics, neither genes nor DNA determine who you are or what you shall do.
Hello everyone, quick biochemistry basics here. So let's talk about genetic code. The DNA acts as a genetic material and encodes all the information required to encode a protein. The specific region of DNA that encodes the genetic information is called a gene, and there can be several genes in the genome. For example, E. coli has roughly 4,500 genes, while humans have 30,000 genes. It's usually by the process of transcription, the genetic information in the DNA flows in RNA, also known as the messenger RNA or mRNA. The information contained in mRNA is further used by the translational machinery to make proteins. This flow of information from DNA to RNA and protein is also known as central dogma of molecular biology. This was given by Francis Crick in the year 1958. The genetic code. Genetic code is a particular sequence of nucleotide that tells which amino acids are to be linked together to form a protein. The genetic code consists of 64 triplets of nucleotides that codes for 20 amino acids. Each of these triplets are known as codons. Now, as there are 64 codons and only 20 amino acids, a single amino acid can be coded by more than one codon. This property of genetic code is known as degeneracy. For example, amino acid proline can be coded by CCU, CCC, CCA, and CCG. However, a single codon will always code for a single amino acid. For example, CCU always codes for proline. AUG always codes for methionine. This property of genetic code is called non-ambiguity. The genetic code is non-overlapping, which means after reading one triplet, the reading frame shifts over the other three nucleotides. The genetic code is commaless, which means there is no extra nucleotide between the codons. The genetic code has start and stop codons. AUG is usually a start codon that codes for methionine, and stop codons are UAA, UAG, and UGAA. The stop codons are also known as nonsense codons as they do not code for any amino acid. The genetic code is nearly universal, which means it is same in humans, bacteria, plants, and amphibians. However, there are some exceptions in mitochondrial genome, single-cell eukaryotes, and some bacteria. The first two bases of the genetic code are same, while at the third position, the base may wobble.